Okay, so what we will see now, we will start with meiosis 1, okay, which is very much important. So meiosis 1, you know, it is important. Meiosis 1 is important, and meiosis 1 is, again, you know that it is divided into karyokinesis and cytokinesis. Karyokinesis 1, cytokinesis 1. And in karyokinesis 1, again, you know that it is divided into what? Prophase 1, metaphase 1, yes, anaphase 1, and then telophase 1. Right? So see here, what we need to understand here is that, see, even if it is mitosis or meiosis, these stages, what actually happen in these stages are same only. Like in prophase, we know that in mitosis what all happened, because in metaphase a chromosome should be at the center. In prophase only what will happen? Condensation of DNA will start. It will form chromosome. Spindle fiber formation will start in prophase, and nuclear envelope will start disintegrating. Nucleolus will start disintegrating. All these things are happening in prophase 1. Same thing will happen here also. In this prophase also same thing will happen. But here we have another divisions. As I told you, we have leptotin, zygotine, pachytine, diplotin, and dikinesis. That is what we are going to discuss now. But just as an overview, you should know what are the concepts, what is actual concept, and what actually happens here. In metaphase, the condensed chromosome, the condensation started in prophase, and during metaphase it will come at the equator. And who will hold them at the center? It is done by spindle fiber. Spindle fiber will support that chromosome to be at the center. In anaphase, what will happen? Splitting. In case of mitosis, anaphase, centromere was split. But here that is not the case. That is the difference. Here we will discuss that. Why it is not, why it is not centromere, why it will not, separated, or why it is not pulled towards the pole. Centromere is not cut here. Why? That you know very well, because it is not equational division, it is reduction. Meiosis 1 is reduction. Okay, we will discuss that. So, but what is anaphase? What is the main feature of anaphase? Chromosomes, they will divide. Then here in telophase, they are towards the pole. Telo means end, and they are carried towards the end. So, these is same for even mitosis, for even meiosis. Only thing what actually the concept is same. And what is the difference that we will see now? Okay. First of all, before talking about all these three, we need to talk about, or we need to discuss about prophase 1. Because in prophase 1, we have different stages. Which are the different stages in prophase 1? Leptotin. Right? And then we have zygotine. Then we have pachytine or pachytine. And then we have diplotin. And the last one is diakinesis. Once all these things are done, then only what will happen? Okay? Then only cytokinesis 1 will happen, and then meiosis 1 is over. Okay. So what actually happens in leptotin also? I'll rub that also. We'll start in detail about meiosis 1. I hope you have taken down. You can pause and take. Okay. So the thing is that in prophase 1, we have these other stages, right? Leptotin, zygotine, and all those things. But we will see the blueprint also. What is the blueprint here? The blueprint is, this is the cell, and I told you if it is human, it is 46. But I am taking an easy example. If I take example of few chromosome, it becomes easy for you. So I am taking example of chromosome 4. So in first meiosis, it will form 2 cells with 2 and 2. This is meiosis 1. Clear? This is the blueprint for meiosis 1. I'm not showing you meiosis 2 here because we are discussing only about meiosis 1. Now, see here, try to understand. Prophase Prophase. I told you prophase is a preparatory phase for which phase? It is a preparatory phase for metaphase 1. Prophase 1 is a preparatory phase for metaphase 1. So what are the main thing what should happen here? I'll write all those points here, which are the main characters of prophase. First of all, in prophase, I'll just writing it here and here. Okay. The first thing what should happen is condensation of DNA. Condensation of DNA. Why it should condense? To form what? To form chromosomes. This is the main point of prophase. Another thing is that as it is getting condensed, what will happen? Next is metaphase. Their spindle fiber should be there. Then where it will start forming? It will start forming here. So spindle fiber formation will start. Right? Again, another 2 points. Third thing, what should happen? Because DNA is getting it is condensed and it's forming chromosome, space is required. And for that reason, if nucleus envelope is there will be no place for that chromosome to move. So nuclear envelope disintegrates. Correct? Then as soon as nuclear envelope will start disintegrating and DNA is condensing into chromosome, then obviously another thing which will disintegrate is nucleolus. Nucleolus disintegration. Right? Okay, so these are the 4 points which are the main points of prophase and which actually should happen from the start till the end of prophase. But since prophase is divided into all these sub-stages, then all these 4 points, what I have written here, all these 4 things should happen from the start of the leptotype till the end of the dikinesis. From the start of leptotype till the end of dikinesis, all these things should be done. Are you understanding this? Okay, fine. I hope you have understood. If you want, you can take down. Fine. So, see here, we will discuss in detail now about all the stages. Fine. So, first let us start with leptotype. And this blueprint, I think you have written somewhere in the corner. I will be rubbing this. Remember, from 4 we have to form 2. It will take time. However, I am going to explain only these stages. Okay. Now, what will happen in leptotype? Our cell has how many? I am talking about leptotype, okay? This is the cell and this is the nucleus. Ishtu dot nucleus virala. Just I am showing it for you to understand. Okay, now how many chromosomes we have taken as an example? 4, which should form 2 and 12. This is a blueprint now for meiosis 1. Okay, so 4 chromosomes. All the 4 chromosomes will not be the same. Why they are not same? Can you think about it? See here, 4 chromosomes. I mean, before this leptotype, before the cell undergoes this prophase 1, okay, the cell will undergo which phase? Interphase. And in interphase, G1, yes, G2, all those things. So in interphase, how will be the cell? The cell along with the nucleus, the chromatin material is like this, no? This is how you will find. This is nucleus envelope, this is chromatin, this is nucleolus. This is how in interphase. This is in interphase stage. After interphase, okay, it will enter meiosis 1, karyokinesis 1 in that leptotype 1. So this starts condensing. So what is the first point of prophase? Condensation of DNA is the first point, and that started over here only. Where? In the leptotin. Andre thin fiber tara thread tara iruhanta chromosome, DNA, it starts condensing to form what? To form chromosome, alva? It will start here. So what will happen? You will be able to see the what you can say, the DNA heng kansutandre illi. You will be able to see the DNA like this. Compaction has started. Compaction start agide. Still it is looking like a thin thread only, no? Then we have, okay, I am showing 4 chromosomes, so I will be showing 4 only. Then we have like this. Like this. Chromosome in the stage of leptotin, they are not at chromosomes. Condensation have just now started. So because of that condensation have started, you know that DNA condensation means what? Histone and DNA will wrap around the histone. DNA will wrap around the histone to form chromosome. Because it has initiated in leptotin, what you will see is that because of the histone and all those things, you will be able to see this beaded type structure. Like this. It has started condensation, no? That is why you will start seeing this type of structure. Like this. And here also, this also. How you are able to see this thing? It is, sorry. This is how you will be able to see the chromosome at leptotin, because initiation of compaction of chromosome have started. Sorry, DNA have started. So that is why it looks like this. Hang kansutta andre ondra beads tara. You will start seeing long chromosomes tight agilla. They are not compacted. They are not condensed here still. They are long chromosomes. So I will be writing the characters of leptotin here. You will be able to see the nucleolus as well here. So in leptotin, what is the concept of leptotin? Here the characters are like this: DNA condensation, or you can say compaction, right? And that is why it looks long, and you can see dot tara kansutalla. So already we have discussed about that. Chromomeres, DNA histone wrap adaga it looks like beads on string anta avagale helide nanu munche ಒಂದು ವಿಡಿಯೋದಲ್ಲಿ. So it looks long in leptotin and beaded. This is important. Long beaded chromosome. We cannot call it chromosome still, but still long and beaded. So leptotin is not a big deal. Very easy it is. So it has started with condensation of chromosome. Innu ivella agbeku. We will see ellelledella agutte anta. First it will start with this in leptotin. So DNA compaction, it is compaction. Okay, COMPACTION. Long beaded chromosome you are able to see in this leptotin. Now the question arises, why I have shown different colored chromosome? DNA will have colors. I am not talking about that. Actually, why I have shown it like this, because I told you already that this cell which is undergoing meiosis, it is a diploid cell. Half set of chromosome is from father, half set of chromosome is from mother. Alva? So we have maternal and paternal chromosomes here. So I have shown this red color ones are anything. You can take it as maternal. For example, red, let us consider it as maternal chromosomes, and blue we will consider it as paternal chromosome. Okay, so because I have taken example of 4 chromosomes, so this is only 4 chromosomes I have shown. In human beings, how many will be there here? 46 will be there. 23 chromosomes with red, 23 chromosomes with blue we need to draw, and that is not easy for us. So we have taken example of 4 chromosomes, but the concept is same. Please relate it. Eshtu irutte red 23, blue will be 23. Clear? So this is leptotin. And one more thing you can understand here. Now already I have discussed about homologous pair. No, I told you the chromosomes will have their pair. The maternal chromosomes will have their pair in paternal. And that thing you can see here. See, this one which I have done here, this pair of this is this one. How you can pair? How you can understand? They both are metacentric. And if you see these two, they both are same. Why? Because you can see their centromere region is little bit away from the center. Right? There are so many things will be there in human cells. But because I have taken 4, it is easy to show me like this. So I have done like this. So the pair of this one is this. These two are homologous pair and these two are homologous pair. Okay? So this is how you can say that homologous chromosomes are present in the diploid cell. But whatever is the thing, what is the 2 important points of leptotein? DNA compaction will start. Because compaction has started, DNA looks like beaded chromosome. It looks like beaded string. So long beaded chromosome. Take down this much. Fine. Now, okay. Now, still the nuclear envelope is there. Still the nucleolus also is present. Next is zygote. What happens in the zygote? Okay, we will see zygote. Now, DNA compaction is still continuing, even if it is zygote. DNA compaction complete, prophase complete agovargo, DNA compaction will be done. Fine. Now, what actually happens in zygote is that, now, because this and this are same and this are same, they are homologous pair. In zygote, what will happen is pairing. I'll show you. Note down this one. You also use different pens so that you will understand easily. Like this only. Now coming to zygote. What will happen in zygote? So, zygote. Let us consider zygote. See, this is a cell. Still the nuclear envelope also is there. In zygote, the main character of zygote is homologous pairing. Pairing of homologous chromosome. That means what I can show is that the metacentric chromosomes what I had done and this pair with the maternal chromosome and the one which was like this, it will pair with the maternal or the paternal, whatever, like this, right? So red is maternal, blue is paternal. What you just understand here is they pair with each other. The homologous pair come near to each other. In the leptotein, what you saw, they all were in haphazard manner. They were not paired. But now you can see they start pairing. If you relate it to human cell, then all the 46 chromosomes were there. 46 chromosomes are there in human cell. 23 pairs are formed, right? From first, second, third, like that. 23 pairs are formed in which stage? Zygote, right? Now, why this pairing has started? Why it will attract to each other? Why they are together? This homologous pairing, yak agutte? Because when they come near to each other and they pair, this process is called pairing, right? There is another name. Pairing is called synapsis. The pairing process of pairing is called synapsis. Andre they just come together. They just remain together anta helbodu. But why they are remaining together? Why they attracted each other? Because they had a complex here. For synapses to form, what is important is there is a complex called synaptonemal complex. Okay, this will be not in this form actually. It is just like a liquid form. It will be present in the liquid form. This is called, the green colored substance what I have done is called synaptonemal complex. So, what you will find here is synaptonemal complex. What is the synaptonemal complex? This is a chemical substance which actually holds both the paternal and maternal pairs, homologous pair together. Why they are together? Because they have an attraction between them. What is that attraction? Is the synaptonemal complex. That is why they came near to each other. Now you can see them together. Correct? Now, so here, these are the important points of zygoting. Homologous pairing will happen. That process is called synapsis. Why synapsis happen? Because of synaptonemal complex. Now if you see, this is one pair and this is one pair. Now this is called bivalent. What do we call it as? Bivalent. And this is also one bivalent. This is also one bivalent. Now don't get confused with the term bivalent. I'll tell you what is bivalent. Bivalent means two chromosomes together. Bi means what? Two. How many chromosomes? Two chromosomes. Okay. Some students will still get confused because even this is chromosome. Like this. And even this is chromosome. So now if you consider in that way, 1, 2, 3, 4, we'll get confused with all that. So better to remember, we call it as bivalent also. We also call it as tetrad. Still you got confused? Bivalent means what? Remember, bivalent means bi. Bi means two. What two? Centromere. So, I hope this much you have taken down. Homologous pairing, synapsis, synaptonemal complex. Please take down that much. Pause and then please take down. And now I am explaining you bivalent. Okay? Now, how many bivalents are you able to see here? Two bivalents. What does a single bivalent means? Means bivalent means, I'll show you, same type of chromosomes, they have come together. Why? Because of the synaptonemal complex. Now these two chromosomes, they have how many centromeres? Two centromere. Because they have two centromere, we call them bivalent. Bivalent because two centromere are found. So this is one bivalent because two centromere. This is one bivalent because of two centromere. And then we also call it as tetrad. I told you we also call it as tetrad. Why tetrad? Because they are called tetrad because how many chromatids are there? This is one chromatid, this is one chromatid, this is one chromatid and this is one chromatid. How many chromatid? 4. So, because of 4 chromatids are there, we call it as, because of 4 chromatids, we call it as tetrad. I hope it is clear for you people. Tetrad. But, one more thing. So, one bivalent will have 2 centromere, means 2 complete chromosomes, and 4 chromatids. We call it as bivalent because it has 2 centromere. We call it as tetrad because it has 4 chromatids. And in this cell, how many bivalents you saw? This is one bivalent, this is one bivalent. Clear? Zygote is very much important because of synapsis, because of the pairing of homologous chromosomes. I hope you understood this much. Take down this much. Okay, fine. So I hope you understood zygote. Let us move on to pachytene or pachytene, whatever. You can remember this very easily in the form of a story. Okay, fine. We will discuss that. Now we have completed leptotene. We have completed zygote. Now let us see what do you mean by pachytene. Now in pachytene, again let us draw the cell. still the nuclear envelope is intact. Still the nucleolus also is intact. Fine. Now what happens in patchy chain? Already we have seen that we have already got chromosomes together, right? Already we have pairing done. Yeah, they have their partners with them now. Which color I had used, I forgot that, only blue I guess. Okay, they are together. Why they are together? Because of synaptonemal complex, right? Because of what? Because of the synaptonemal complex, which was, let us consider the synaptonemal complex in green. Okay, fine. Now what happens in patchy chain? Patch formation. Okay, you remember it like that. Patchy chain means patching. What is getting patched? I told you already, we have discussed. We call it as, very important point in patchy chain is that the first thing is crossing over. What happens in patchy chain is crossing over. What is crossing over? I told you, when two non-sister chromatids. Now this chromatid and this chromatid, the chromatids of these two paternal and maternal chromosomes are called non-sister. Here also they are called non-sister. So there is a crossing over between non-sister chromatids. I'll show you how. For example, they are together now. You can see there is a crossing. Because they are together, what will happen? They cross. Like this. And here also the same thing will happen. Same thing with this also. This side also it will cross. Like this. And this will cross with this. Yes? And same thing with this. What happens here is that you will see crossing over like this. Like this. They crossed. So, what you can see in this patchy chain is crossing over. I told you crossing over is nothing but crossing of non-sister chromatids. Because they are together, they cross each other. This crossing over leads to what? When they cross, you can see there is thick, what you can see? Thick area, you will find it. Wherever they have crossed, there you will find this thick area. So this thick area is called recombination nodule. Because the two fragments have crossed here, this area looks very thick and that is called recombination nodule. So another important point in patchy chain is what you will find? You will find recombination nodule. Now what is the importance of this nodule? This nodule has an enzyme called recombinase. This is important. What it has? It has enzyme called recombinase. Now what does this enzyme does? You know that crossing over leads to what? It leads to recombination of genes. That means try to understand, because of this enzyme, recombinase enzyme, the patches will exchange their segments. Wherever they have crossed, this particular paternal chromosome will lose its segment and it is attached to the maternal one, and maternal will lose its segment and is attached to paternal. Already we have discussed crossing over. Please watch that video if you are not understanding it here, the previous one. Okay. So, because of this enzyme only, then what this enzyme has, so that it is cutting the segment and helping it to attach to other one. It has two enzymes which are called as restriction enzymes. Restriction endonuclease you might have heard about, which actually helps in cleaving the segments of DNA. So that, and it also has ligase. Ligase will help in joining. So what restriction endonuclease will do, it will cut one segment of paternal and then ligase will attach it to the other one. So this type of exchange of segments will happen between these two and these two and these two. Understanding? So this is what happens in patchy chain. I hope this is clear. Okay, so this much you take down.